Thank you. Okay, so my name's Rachel Mills. I'm the Senior Vice President Academic here at King's. That's sort of the provost role. I'm Rachel's boss. that's how she's introduced me. But I'm here on behalf of the Vice Chancellor, Kshitij Kapoor, who couldn't be with us tonight. And he likes to attend these inaugural events, but tonight he couldn't be here, I'm afraid, so you've got me. I'm just going to say a few words of introduction before Rachel gets going. And it's great to have colleagues, family, friends, as well as people from across King's and elsewhere, and from the maths school here tonight. A special extra lesson. This is a very special inaugural lecture because it's been given by the Dean in the Faculty of Natural, Mathematical and Engineering Sciences lecture series that aims to celebrate the new professors that come into the faculty. I asked the faculty to allow Rachel to leapfrog the waiting list because I thought it was really important that we hear from her as the new dean, especially coming in new to King's, and so we get to hear her story, hear her journey, hear her passion, and hear what excites her. Okay, these are wonderful opportunities to share the remarkable career trajectories of all of our professors here at King's. And so Rachel joined King's in January 2024, as I said, the executive dean of this faculty. But more importantly for tonight, a professor of mathematical biology. And you'll hear this is a recurrent theme, I think, tonight, that Rachel uses her math to apply it to lots and lots of different disciplines, specifically biological disciplines, but you'll see the huge range of that. She is a true interdisciplinary scientist. And interdisciplinarity is something we're really proud of here at King's. It's something we're really trying to push. It's something that we're investing in. Interdisciplinary science in all of its facets is something that we think is where the exciting things are happening, where the exciting discoveries are made, about finding solutions to those global tricky problems that we're all really working on. So I'm going to be really excited to hear how she's pioneering the application of mathematical principles to understand spatial and temporal dynamics of biological systems, solve health challenges, and answer fundamental questions. This includes various things. I'm going to keep talking until people come and sit down. So here's some sort of foregrounding of what you're going to hear. You're going to hear about bacterial chemotaxis. I had to Google what that meant, but I'm sure she's going to tell us. We're going to hear about cancer cell motility. We're going to hear about phytoplankton behavior in turbulent environments, a subject close to my heart, as well as exploring cell signaling pathways and drug transport. So a huge range of things. Rachel's passionate and an advocate for STEM education in the broader sense. She served as the 2022 president of the mathematics section of the British Science Association. She was a trustee of the University of Liverpool's Maths School, which was the first specialist 16 to 19-year-old maths school in the north of England, something, of course, that's grown from the King's Maths School down here, which we're going to hear about a bit, I'm sure. So if we're all here and we've got a seat, anyone else that needs to come in while I'm talking? I think we're ready. Please join me now to welcome Professor Rachel Bearon to present her inaugural lecture.
Thank you. Wow. I was going to start by saying thank you, and it's lovely to see you all, friends, family, colleagues, new and old. But I'm also thinking I should start by saying, goodness, I hope I deliver on all those things I'm going to talk about. We'll see how much I get through, but it really is a pleasure to be here. Thank you so much for coming. So what I'm going to talk about today, to start off with, I want to get across the message about maths being both beautiful and powerful. I'm then going to talk a little bit about my journey as a mathematician and highlight some of the opportunities I've had and some of the great collaborations that I've enjoyed taking part in. I'll then go into a little bit more depth about a couple of research examples to illustrate some of these collaborations in the area of math biology. And finally, touch on a little bit of collaborations beyond science. So a collaboration I had with some composers and looking at reflecting a little bit on what it means to be creative. So maths is beautiful. And it's a really intrinsic part of being human. It's got a long history. For example, we can go back to the ancient Greeks and Euclid several thousand years ago and see the beautiful geometry that was written about at that time. Now, as a kind of personal anecdote, I bought a copy of Euclid's Elements, you can still buy it on Amazon, last year to share with my oldest son, who's interested in wanting to study classics. He's studying maths and classics at A-level. And what's so great about this book is you can still read it and it's still really fresh. And the geometry that Euclid wrote about several thousand years ago is still true. And maths is something which is really exciting because of that. So beautiful, so elegant. So that I've gone aside, I've got like different groups around the room. I can spot like the maths corner and the physics corner, and the maths corner lit up at that point. You get what I mean. So that beauty and elegance really captured the popular imagination, I think, with an example of that is with Fermat's last theorem. So it's such an elegant statement. If you have x, y, and z being positive integers, the only solution of this is when n is an integer, is when n is 0, 1, or 2. Now, many of you will be familiar with the 2 case, things like Pythagoras' theorem, when n is 1, simple lines, simple equations. But the fact that there's no solution when n's 3 or 4 or 5 is really, really fascinating. And I wanted to illustrate that it's a real joy of mathematics as well. So I don't know how well you can see this picture, but this photo of Andrew Wiles displaying his proof of this at the Isaac Newton Institute in Cambridge. And there is such joy at that sense of discovery and sharing that ability to solve that problem. And it's hugely complex, the proof, but a very simple and elegant statement, which really, I think, captures some of the beauty of maths. It can also be incredibly visually beautiful. This is a figure generated not from Gen AI, but generated from a colleague of mine, Lassie Remper, who is a colleague in Liverpool working on complex dynamics. And this is some work with Julia sets, which generates this rather attractive figure there. It's visual beauty, but it's also beauty and elegance in abstract thought. I think Hardy captures this very well in a quote from his book on the mathematician's apology about the importance of beauty in mathematics. Now, that aspect of beautiful but also intrinsic part of being a human, as a lecturer, we're always encouraged to be active learning. So here's a quick exercise for you. Who's done a puzzle this last week, like a Sudoku or one of these, puzzle games or things? There's a few hands. There's quite a few people who like to do the odd puzzle. And I have a bit of a guilty secret on puzzles. So when I was a primary school child, I used to love puzzles. My mum's chuckling away, we'd always go on train journeys and I'd always have logic puzzles and loved doing those as primary school kid age. And then I kind of went off it when I was secondary school, and it was a bit beneath me, and these things, a bit of a waste of time. And no, you don't look at those. But I have to admit that COVID hit, lockdown hit, and my family we were kind of so into these puzzles, and now it's that race on a Saturday, who can get the puzzles. So this aspect of puzzle solving, maybe not true mathematics, but I think it's that aspect of problem solving and intrinsic beauty of maths that appeals to huge numbers of the population, even people who wouldn't necessarily call themselves mathematicians. So mathematics as beauty and an intrinsic part of being human, but it's also incredibly powerful. And I've chosen two examples, which Rachel's already mentioned. Two organisations very close to my heart have had news reports about the power of mathematics very recently. So the Academy for Mathematical Sciences published a report just a couple of weeks ago with some new analysis about the contribution to the economy, very timely after the recent budget. So the GVA, directly associated with mathematical sciences, accounts for 20% of the UK's total GVA. This is incredibly powerful in terms of the economic impact of mathematical sciences. So that's on the economic money figures side of things, but I also love the stories of individuals. So also just in the last few weeks, there was a piece from the King's Maths School which appeared in the Times, and one of the graduates from the King's Maths School was featured there. I think a couple actually were, and highlighting what a great training and background mathematics was, and the wonderful things that these young people were going on to in terms of STEM careers. So both the economic financial contribution, but also the stories and that life-changing opportunity there is with maths. Maths has real-world impact and it's incredibly powerful. So, my journey. There's a few pictures around. There's a few people you might spot in the audience as well. I'm talking about my journey of collaboration and opportunities. So I was an undergraduate at Cambridge, and the first kind of collaborator and person I'm highlighting here is one of my very close and dear friends, Sarah Dance. Some of you might know she's a professor in maths at Reading, joint between maths and meteorology. And we work very closely together as undergraduate students. So after every morning's lectures, we'd be going back and trying to figure out what on earth was going on and talking to each other and working together. And that working together and collaborating was really important to me as a student. After my undergraduate, I did a PhD in Cambridge with Tim Pedley, and it's absolutely lovely that Tim's here today. The PhD was on run and tumble chemotaxis in a shear flow. So what was that about? It was about modelling swimming bugs and how they sense and respond to chemical cues and how their motion is modified in flow fields. So how do they sense gradients? So this is particularly applicable to things like bacteria, marine bacteria, searching nutrients and so forth. And I really enjoyed my PhD. I have to say, I'm sure I won't embarrass him, but he was a great supervisor. He set us up with really interesting problems to tackle, and then gave us lots of freedom to explore, take risks, make mistakes, and that's okay, and have a great experience really pushing ourselves as students. We also had a weekly biomaths, so the research group, our PhDs, postdocs, visitors coming from all over the world would meet up each week, we'd talk about our research, we'd talk about other people's research. And I think that community is a really important aspect of doing research mathematics and many other research activities that we do in our faculty. So after that, home in mathematics, I went off to the States, and I did a postdoc work at the University of Washington in Seattle, and I was actually based in a biological oceanography department there. I naively thought I'd go in as a mathematician. I'd develop some models of swimming algae and would be able to predict algal blooms and solve all the world's problems. 4 years later, I was still kind of carrying around tanks of algae and growing them and filming them and, doing some stuff on it. And it was an amazing experience, that interdisciplinary research, really understanding what experimentalists do, how challenging it is sometimes, having some really fruitful conversations to try and pin down what is the interesting science question that we have here. I'm still very much a mathematician at heart all the way through. So I was really, really glad to be appointed as a lecturer back at the University of Liverpool in the maths department there. I'm not going to be talking too much about my experience as a lecturer in the teaching and the education, but just to highlight here that this has always been a really important part of why I love being in higher education. Higher education wouldn't be anything without students. I was saying this to people earlier. We had our meeting, our staff forum, and we did talk NSS. I'm looking at Rachel here very much. We had a really good constructive meeting about NSS action plans. And what I was saying there is, this is in all of our interests if we're in universities, how we, what the experiences that our students have. It's so important to us. For me, it's a huge privilege to journey alongside students and be alongside them as they have that journey of discovery. And I loved lecturing. I love being involved with students, and was very happy. Had a very happy time at Liverpool. Just picking up a couple of research things along the way. My background, as I said, was in biological fluid dynamics, looking at swimming microorganisms in flow environments. But I always enjoyed the opportunity to take part in new collaborations. So I was involved in a really interesting collaboration, systems biology collaboration with a great imaging lab. They did a lot of single cell imaging, looking at protein dynamics in cells. They were very open to have me as a mathematician involved in those projects. And that led on to us forming the Liverpool Centre for Maths and Healthcare, and a couple of the more recent projects that I'll touch on a little bit today. It's great, Smita's here, one of my postdocs, one of my recent postdocs, who's just joined at Imperial, continued some of the work that I did right back from my PhD, looking at those swimming bugs in flow fields, still many open questions. and also some work that another great postdoc of mine, Sean Edwards, did on looking at drug transport through the oral mucosa. And if anyone is looking for a postdoc, there is a postdoc connected with that project live at the moment in the Maths department. And then I'm here at King's. So you might be interested to know, well, why the move to King's? I've said I had a great time at Liverpool. I've really enjoyed my time at Liverpool. Well, I think around 2020, or just before that, I started taking on more leadership roles. And I was head of department for the last few years I was at Liverpool. And one of the things I really enjoyed and found really rewarding was that sharing the journey with others. And as a lecturer, that sharing the journey with students was really important. As a head of department, I was able to share the journey with academics. I was able to look at how we support and encourage and enable academics to do amazing things. And that opportunity is such a privilege because, you know, academics are such a talented bunch, have so many great ideas. We've got so much opportunity to do great things. And I found that really, really rewarding. So the position at King's kind of came up and I thought, oh, that sounds interesting. Not only do I have kind of leadership in maths, but there's the opportunity to lead a faculty where we have engineering and physics and chemistry and informatics. What exciting things do they do? It was a really exciting opportunity to have that breadth. It was really exciting, the thought of being in London actually as well. So in work that I've been involved in with the Academy, being close to that centre of government was very interesting to me. And I think the final thing that really interested me was the strength of the leadership that there was. So there was a real commitment to some really exciting opportunities. We've got the quad, the engineering quad in our faculty. I mean, the vision to establish, a brand new engineering facility right in the heart of King's. The vision to establish KISS, King's Interdisciplinary Science, and see those opportunities was very exciting to me. So the opportunity to sit around that leadership table to both learn from it and to contribute it was great. And having arrived, it's not disappointed. The faculty is incredible. I go from having a lunch with an inorganic chemist to talking to someone about robots, to hearing about someone doing string theory. There's so much exciting and interesting things in. So thank you for hiring me, and I'm very privileged to be here. So let me give you a couple of research examples, starting with the swimming bugs. So one of the things I found very interesting is the mathematics associated with the motion of fluids around ocean liners is the same as that associated around swimming bugs. Navier-Stokes equations can be used for describing both of them. And really the key difference and the key non-dimensional parameter is the Reynolds number, distinguishing the two different dynamics. And the Reynolds number is a measure of how the relative strength of the inertial forces to the viscous forces. So when we're dealing with bacteria, which are tiny, the Reynolds number is very small. They're small, they move quite slowly, and we have really they have really no sort of inertia. So if they're moving and they stop swimming, they just stop. There's no coasting. And some of the nice things about this low Reynolds number world is that they can do some really interesting analysis, but some of it's quite counterintuitive how they move. But there's been quite a bit of study about this, and I remember seeing this work about bacteria the first time, being fascinated. So these are kind of some of the smallest natural micro swimmers. They have a body, some semi-rigid helix that spins around as a tail, pushing them through the water. And if they want to find their way through an environment, they kind of do this random walk whereby they have multiple flagella. If they all rotate in one direction, they swim in a nice smooth path. If one of the flagella flips in the other sense, they kind of tumble around and they can sort themselves out and go off in some new direction, and they can do this in a biased way. And there's some movies there showing the swimming behaviour. And this was the area of things I worked on in my PhD. Now, sometime after my PhD, and this kind of illustrates the sort of process of research, you work something out, and then you see some experiments which don't agree with what your theory says, and so you have to move on your theory. So this paper was an example where it was like, oh, that doesn't agree with what I thought. So let me just talk you through these results. So we've got a microfluidic chamber. There's fluids going through that path, and we have a bunch of bacteria in there. And starting here, the bacteria, the little grey dots, are scattered all around, and this is the sort of distribution function of the bacteria, so fairly uniform across the channel. Turn on the shear, and the green dotted line gives the distribution, and you see there's a kind of depletion from the centre line. Now, you might say, well, is this kind of sort of centrifugal, force sending them out to the edges? No, it's very much to do with the swimming behaviour, because if you put dead cells in, you don't get that. Can we describe this? Can we model that? The theory that I'm doing for my PhD wouldn't be able to capture that. So what is the right mathematical model? So I'm bringing this in because I'm sort of working on the principle, I think it's kind of about A-level maths level. We'll be following this through. So how do we describe what the swimming bugs do? They have a position that changes with time, and we kind of know that the rate of change of position is given by the velocity, the velocity of the object. The velocity of the swimmer is made up of two parts. One is the fact that it swims, and the other is that the fluid advects it around. So this is a swimming speed, a direction, and an advection. And then we have to say, well, how does the direction change? How does this vector p, which says what way you're pointing, change with time? Thankfully, we can go to some classical mathematics from 100 years ago, and we can write down how it changes with time. It's called a Jeffrey orbit, and I've just illustrated it with a little movie there. And what you find quite interesting in shear flow, if you have an ellipsoid, there's a closed form, a nice simple equation for it, but you get non-uniform rotation. So this would be an equation describing the rate of change of the angle given by a rotation, by the angular velocity, and it's non-uniform. Now, there needs to be some randomness in order to generate this phenomena. So if I'm talking to mathematicians, I'd say we extend and go to a stochastic differential equation. If I'm talking to engineers, I might say, well, we just kind of separate the differential equation out and shove on some white noise. Either way, depending on who I'm talking to, you get an equation like this, which can be shown to be well-founded. Which tells us how a small amount of theta changes due to the rotation and due to some white noise. And then we can simulate this. So I just got a quick and dirty movie simulating this. So we've put in Poisson flow, and I've done double periodic Poisson flow, so we don't have to worry about any boundaries. And all the little spots are the little swimmers, the little ellipsoid swimmers doing some random walk. And the paths show an example swimmer where it's going. And then what we've got here is a snapshot, if we put loads of little bugs in here, what their distribution looks like after some time. And what we see is that where we're in the region here, where the shear is minimal, so there's very little change in flow speed, we get a depletion of cells. Whereas where the shear is maximal, where the direction changes, the velocity of the fluid changes direction, and we get big changes, big shear, you get accumulation. So we can capture the phenomena using this model. Can we understand it? Well, it's useful to have a little bit more maths to see if we can understand it. So I think this is about as mathsy as the slide gets. So enjoy, mathematicians, and the rest of you will move on quickly. So probability distributions are a really good way of capturing the distribution and the density of cells. And that stochastic differential equation can directly map onto a Fokker-Planck or Smoluchowski equation, or if you're a fluid dynamicist, you think of this as a conservation equation, or an engineer's conservation equation for the density or the probability distribution. And we've got a few things balancing in that probability distribution. We've got the first term represents the swimming to non-dimensionalized speed mu. That second term with a beta in there represents the non-uniform rotation, and beta is the shape parameter for the object. And the final number with a 1 over the Peclet number is the randomness. Peclet is typically the non-dimensional comparing the advection to the diffusion. And without any fitting, so just for the experimental data, we get fairly good agreement between the experimental data, the red, and green, which is the full solution of that equation, numerically solved. But to push it a little bit further in understanding, we can do a bit of asymptotics and separate out that distribution function to be the density distribution multiplied by the orientation distribution. And we can capture why we get a depletion in some regions and a peak in others, because the density is inversely proportional to the mean square cross-channel swimming speed. Now, let me try and explain that a little bit more in pictures. So how do we get this high shear trapping? We start off with a rotating bug. Maybe we don't start with a rotating bug, a non-rotating bug. So this is supposed to rotate around and remind us that when we're slender, we have that non-uniform rotation. So if you're slender, you have non-uniform rotation, and when you're in the high shear region, you spend more time swimming horizontally. Whereas in the lower shear regions, you're less affected by this non-uniform rotation, so you can swim up and down a bit more, which means those cells where there's accumulation, the cells are not swimming up and down, so they accumulate. And that explains the mechanism for that trapping in high shear quite comfortably. Also, I thought, so we know I said how science works. You prove something, you work it out, you're all happy until you find something that it doesn't work for. So that was the same here. We did a bit more work looking at how shape varied things. So beta is the shape parameter, 0 is a sphere, beta going to 1 is an infinite long rod. And if we look at that approximate solution, which is the one which explains the mechanism, how we get n being proportional to 1 over VMS, it works beautifully. We get this depletion. However, if we go back to the full probability distribution, we get a non-monotonic distribution, so a double peaked distribution at moderate values of beta, which we couldn't understand at that point. So it was left as a somewhat open question when we were finishing that work off. However, maths is a very collaborative worldwide endeavour. So I was delighted that Ganesh Subramanian, a wonderful mathematician currently based in India, published this some few years later, which captured and was able to explain mathematically what was going on there. So as you increase that Peclet number for moderate values of beta, you can get these double peaked distributions, which, as I say, is a beautiful paper explaining that in quite some depth. And I'll also highlight Smita's work as well, where some of the unusual behaviours that we saw that was in the boundaries are properly explained. So that's some follow-on work about swimming bugs. That was the end of swimming bugs. One more example, research example. Quite a different story. So I think in my introduction, Rachel kindly said I apply maths to lots of different things. Some people might say kind of jack of all trades, but we won't go down there. But I'm always interested in new problems. So this is a problem that came about through some interesting collaborations. And the challenge here is, if you're designing in the pharma industry, if you're designing drugs, how do we know if they work? So traditionally, a lot of experiments have been done on Petri dishes, where you kind of pop the cells in, form a layer, and you put your drug on top. Now, the problem is with this is that cells that are kind of in a nice 2D environment behave very differently to cells that are in tissues in the body. So there's been quite a bit of work, say in the pharma industry and elsewhere, to develop these spheroids or a variety of other microtissue models where the cells are more physiologically realistic, and also you can capture some of the natural gradients that you might see in tissues, gradients in oxygen or other things. So we were working with people who wanted to develop these experimentally and wanted to understand better what was happening inside. So we developed some models for this. And And we work closely with tissue engineers, so we could get some real interesting detail about the experimental system. So this is a picture slice of the spheroid, and we can extract out the blue dots are all the cell nuclei and construct the where the cells are. And then we can really zoom in and understand what was happening like between the cells. So you can zoom in and the gaps between the cells. There's these very, very thin gaps, these intracellular gaps that we can visualize. Now, I hope you don't get too phased by this. I would be able to, I would tell you pause this simulation, so it'll keep going. But let me talk you through the mathematical simulation of this. So we've got our spheroid, our slice of our spheroid up here with all those cells that come from the geometry of the system. And we put the spheroid in a kind of big vat, and then are going to simulate drug being delivered from the outer boundary and being diffusing in and then being taken up by this spheroid test system. And we're really capturing as much as we can about the details of the geometry of the spheroid, geometry of the cells. And the equations we solve are quite simple. We're solving inside every cell at the top. We model the diffusion of the drug and the metabolism, the uptake of the drug. And outside every cell, the drug diffuses. And then one of the key things is understanding the permeability. So at the interface, the cell membrane interface, we model how easy it is for the drug to get across the cell membranes. So the key parameters there is Q being the permeability. And then this is just kind of zooming in to show how this is done. And this is all was simulated numerically once we've got the equations down. And here are some example simulations. These have slightly different boundary conditions. These are examples where rather than having a fixed outer concentration, we deliver a fixed quantity of drug with no flux outer boundary conditions. And this is exploring the impact of the permeability. So Q was how easy it is for a drug to get through the cell membrane. So on the left-hand side, it's really easy for the drug to get through the cell membrane, and the drug just kind of moves in and diffuses through the spheroid. On the right-hand side, it's the opposite. They're like kind of, it has its sort of channels through the spheroid. It can get through the gaps, but it can't very easily get into the cells. And the bottom pictures are kind of comparing, well, what happens if you are a cell right in the center? How do you see the drug? Versus what happens if you're an example cell right on the edge? How do you see the drug? And of course, if it's highly permeable, the drug is absorbed quite rapidly. This is the time scale. Whereas if the drug is quite slow to be absorbed, it takes a long time to get in. And, you know, in between, there's slightly different things happening. Fine, kind of what you expect. What's interesting is if you start pushing the interpretation a little bit further. So in the context of drug design, let's start at the top. So let's start with the case where the cell membrane is quite impermeable to the drug. So in that case, up at the top, it's taking a really long time for drug to get in and attack the tissue, okay? And it takes a really long time, whether you're right on the edge, on the outside of the spheroid, or right in the middle. It just takes a long time. So let's, as a kind of cartoon drug designer, think, well, what we need to do is make the cell more permeable so that the drug can get in. Well, if you make it a little bit more permeable, what then happens is on the outside of the spheroid, lots of the drug will get in and be taken up and metabolized, which is great for, you know, attacking cells on the outside. But of course, the cells right in the center, there's no drug that gets there. So actually, if you're thinking of treating and wanting the drug to get throughout and attack that core of the tissue, this has been detrimental, increasing the permeability. You really need to increase it a lot more, and then you'll be able to get to that point back again where the drug gets through everywhere. So is this slightly counterintuitive? It makes logical sense, but just pushing it in the modelling kind of illustrates how we can tackle some of these questions. So this was done in a fairly theoretical way. It was inspired by our colleagues in the pharma industry, but we weren't able to go back and do sort of actual experiments on looking at drug distributions. So I was really interested when we got a follow-on project. So this was through the NC3R's follow-on project, which we've just got a paper published on last month. I still need to battle with Pure to upload it. I will get that done. And this is an interesting project which was looking at delivering drugs in a slightly different way, which is through a patch attached to the oral mucosa, And what they're wanting to do is explore how the drug gets delivered through that way. So here's a picture of the patch. I think on one of my colleagues' cheek. They used themselves as tests quite a bit. And this is a slice going in of the oral mucosa. So you kind of punch out a little bit from your cheek and you can see the distribution of cells. And there's quite interesting geometry changes as you go from the superficial to the basal layer. Zooming in a little bit further, we could explore the gaps between the cells. And then it got kind of interesting and creative when we were talking to our experimentalists, because we're like, okay, well, how do you measure the gap? You know, the width between the cells, how do you measure that? And they're like, oh, well, we've got these wiggles and and I'm like, hold on a sec, what do you mean you've got these wiggles? That's really going to change the transport. And this was something that, you know, we didn't really know about before. So we characterized these wiggles between the cells, that extracellular space, because that will have effect on how the drug will move through this system. And we did some modeling to look at the distribution of drug in that wiggle. And we did some analysis and some quite simple maths to look at how we could approximate and come up with an effective diffusivity that would be appropriate for an equivalent straight line gap in terms of speed of diffusion. And then we scaled this up and was able to do some tests against experiments. So let me just talk you through the top figure. A is a tissue engineered model of this oral mucosa. So TE, tissue engineered normal oral mucosa, and they put it in a chamber. They have these wonderful things being able to grow bits of tissue, put the drug on top, look at it, how quickly it goes through and can collect it at the bottom, which allows us to parameterize for certain drugs and then use the model to extrapolate for other systems. So taking the tissue engineered model, we can do the maths of identifying the nuclei, some RNA tessellation to kind of construct the cells, put in some spacing based on the detailed TEM, set up the grid for the modeling with the effective gaps, and then set the model going. So here's the simulation regime. This is the detailed bit of modeling that we have of the cell membrane. Sorry, not the cell membrane, the superficial layer, which is the main part we're interested in modeling. Put the drug on the top, it diffuses through. There's quite a bit of this bottom layer, which is quite sort of modeled just there. And we see over time how the drug diffuses. We can get a real, detailed understanding of the geometry, which we then mathematically would want to homogenize upwards so we can do larger tissue samples. And this is some ongoing work. Okay. So that was the second research example. And let me move on to the final few slides. Actually, we've got a little bit of time for this, which is nice. So let me talk about the third collaboration that I want to mention, which is a really interesting and joyful experience. So this is a photo of Emily Howard. She's a professor of composition at Royal Northern College of Music, and she spent some time as a Leverhulme artist in residence in my department at Liverpool. And the picture here is her at an event where they were doing the premiere of this piece, Outlier, performed by Paul Silverthorne, very talented viola player. So let me tell you how this came about. So Emily was as I say, artist in residence in our department, and we enjoyed chatting. I always enjoy chatting to anyone about interesting things. And she was very interested. She comes from a maths and computing background, and she was very interested to learn more of really understanding the maths and the science to be inspired and be interested in what we were doing, what she was doing. So the science project that I taught with her is the last sciencey project I'll talk to you about. This is another spheroid, and this is a spheroid using glioblastoma cells, which is a very aggressive form of brain cancer. Marianne was my PhD student working on that. On the right are the images of the actual cells, and the white dots are kind of the nuclei you can see wiggling around. And on the left is the tracks of individual cells. And we wanted to understand the migration pattern of these cells and the distribution of cells. And often when we're modeling collections of cells, we assume they're all the same. But when we were teasing out some of the statistics about that, it became very clear that actually the cells, there was quite a bit of variation. They weren't all the same. It appeared from the statistics that there were some outlier cells, which is of course really important in understanding cancer with metastasis and those outlier cells, what are they doing? And this was the conversation that we shared and discussed with Emily. And that idea of outliers and behaviours that are outside the norm really intrigued her, and her piece that she composed was built around that. The other thing that I really enjoyed talking with Emily about is the creative process. So what is it that drives us, as scientists, as mathematicians, as engineers? What is it that we say, oh, that's a really interesting problem and puzzle, and I really want to understand that, and how am I going to look at solving that, and how am I going to be creative to find out how to progress this? It really is a creative process, science, in the same way that being a composer or being a poet is a creative process. I don't think it's always kind of recognized, but doing research is about being creative. You're creating things that people didn't know before, and that determination to puzzle through it. So we had a lot in common when we talked about what drove us and where we were coming from and what motivates us to keep going. So that was fascinating. And then to follow on, I had the opportunity to work with one of Anna's PhD students. I love the idea. She's a PhD student who was writing an opera for her PhD. But that was great. So this is a project called Prism 8 Cubed, and it involved 8 composers, 8 scientists. So I was one of the scientists involved, and musicians. And I hope you recognise this picture. This was the picture of the uptake, and so I worked very closely with Anna Appleby, and we're going to hear the piece of music that she wrote. It's about 3 minutes piece of music, and I have to remind myself what the connection is, because when you hear the music, it might not be totally obvious what the connection is. It's a beautiful piece of music. So it's a piece called Uptake. And it consists of 3 miniatures. So the first miniature is the impact of permeability on drug distribution. And we've got piano and bass guitar lines, which actually were inspired by the shape of some of the graphs in one of the figures of the paper. We spent several lunches going through in detail the paper. Second miniature is a little bit more abstract. It's emulating the cell-based models with a simple symmetric continuum model, figure 5. And what's interesting here is we explored the concepts of continuous and discrete functions. So we've got the continuous line of the alto sax melody with discrete moments in the piano and bass guitar parts. And the third miniature, impact of drug lipophilicity, that's the uptake part of the parameter Q, on uptake and metabolism in different regions of the spheroid, is figure 6. In which mathematical modelling yields counterintuitive results at times, and this is particularly useful if you're rolling out to clinical applications where resources are expensive. So expect some counterintuitive results in this final thing. So sit back and enjoy it for 3 minutes, I think the piece is. I hope we're all lined up for this. And I should say, advert, NMES Christmas party, Battle of the Bands, be there, or don't. So let's enjoy this. So that's all from me. Just a final kind of message out there. I hope you've enjoyed hearing a little bit about collaborations and opportunities I've had. And then I often like to end on this sort of final call to action, really, which is about that interdisciplinary working and about that collaborative working. Sometimes it's good to go fast. Sometimes we just need to lock ourselves in a room and get on with it and get something done. But to really achieve some of those big challenges that we need to face and achieve some great things, working together is fantastic to do. Thank you.
Thank you, Rachel. I'm going to now do the vote of thanks. You might want to sit down because I'm going to go on a bit. Otherwise, you have to stand there while I talk about you. But what a wonderful experience. How often do we hear music played during maths lectures? I certainly don't. So thank you for opening my eyes to new connections. And I think that's a theme of what I'm going to say. And I won't go on too long because I know you are all desperate to actually talk to each other and talk to Rachel about her wonderful lecture tonight. But the point of today is to learn much more about Rachel. We've read her biography on the website, or I've seen her CV when we were hiring her, but she has really brought it to life tonight. And that's what these inaugural lectures are about. They're turning those words on the page that we look at as we're doing the hiring process into the real person. And thank you for doing that. You've made me understand more about the beauty of maths the joy of math, something that perhaps not all of us in the room feel all of the time. And we've heard about your guilty secret, that you're doing puzzles, perhaps during meetings, I don't know. And we've certainly seen your passion for the impact of maths on society and on individuals. And I can really feel that today. So let's just talk a little bit about that. It was great to see your timeline that you showed. I really liked that sort of pathway. I first met Rachel, it turns out, I think it was about 2017 in Liverpool. I was an oceanographer at the time. I went up to Liverpool to talk to them about women in leadership roles in science in particular, and Athena Swan submissions. We were in the same room workshopping how we managed to get more women into leadership roles. And who would have thought that we'd both be here at King's in this fantastically diverse organization 7-8 years later. So that was a great start to our, it took me a while to realize where I'd met her before when we were interviewing. But I wanted to say a little bit about the things Rachel hasn't said about the impact she's had since she's come here to King's, and I'm going to embarrass you a bit because I want to say that she's made an extraordinary impact since she started in January 2024. That impact is across both in the faculty, but also across the wider university. And you can see that tonight by who's turned up, who's engaged, who's come from outside the organisation as well. It's her inclusive leadership style and her ability to build those connections that I think comes through really strong today, but it comes through in what I see happening. When I go to the Faculty Executive Board, I can see it. When I join celebratory events in NMES, and they have lots of them, I can see it. And I think that is a truly exceptional characteristic of a leader, is to be able to bring people together and make them feel part of something, and to make things seem a lot simpler than they are. We saw that in the way you described the maths. I think I understand it, but it's the way you bring that to the table as well, that you make the solutions to the problems we're all struggling with seem achievable and seem relatively simple. So I can see you creating that comprehensive plan that provides a supportive and responsive environment in the faculty and across the university, where our students and future students feel valued and empowered and supported. And that's really all we're about. And if we can all do that, we'll be a great place. So she's one of our 9 executive deans here at King's. We have 9 faculties, which is quite a lot. And she's a core member of that team already. Only been here several months, and she really plays an important point. One thing she didn't say was before she even got here, she was writing a proposal with Professor Ajay Shah from the Faculty of Life Sciences and Medicine, and that proposal was successful. And Ajay tells me, he couldn't be here tonight, unfortunately, that she played an absolutely crucial role in shaping the project, understanding the therapeutic potential of phosphoglycerate mutase 2 in the stressed heart. I think it's something to do with all those cells moving about. So Ajay says thank you, and that's really impressive that before she even got here, she's writing proposals and they're getting funded. And so I think we've seen some really good examples of how to make maths accessible, how to turn something that's quite incomprehensible to a mere oceanographer to something I sort of understand, and that is a real talent. And I think all of us could learn from that. I love the bit where you talked about how you explain things differently to engineers, to mathematicians, and to physicists. I heard the laugh here because it's that ability to turn concepts into relevant language for different disciplines that is really, really crucial. And more of us need to learn that. I am embarrassing, I can tell. More of us need to learn that in terms of how to communicate across disciplinary boundaries, because we will never achieve our interdisciplinary aspirations unless we can communicate across those boundaries. So I think we could be more Rachel as we try and do that. And then I particularly like the way you placed your research, your theoretical constructs into that sort of corpus of here's the experimental data that I support, and here's the people that took my ideas and turned it into something else. And again, we need to remember that as scientists when we're talking about our impact, put it into that context. You did that beautifully tonight. So I'm going to wrap up and just say, fantastic lecture. I've learned lots about you, despite having worked with you over the last, whatever it is, 11 months. And I'm sure everybody in the room feels the same way. So let's join together and clap again as we end this evening to thank Rachel for her amazing inaugural lecture. We've learned a lot. We've learned how mathematicians contribute to society, to the university, to the future. And please come and join us for some drinks. And I think we might even feed you a little bit. And we can raise a toast to Rachel outside. So congratulations on the lecture, Rachel.